Session 1 on the topic In Silico Studies, Molecular Docking by Honorable Dr. Shankar Alikav Sir. Sir is the HOD, Department of Pharmaceutical Chemistry, KLE Belgavi. So firstly, I would like to welcome Honorable Dr. Shankar Alikav Sir for the today's session. Now I would also like to welcome Honorable Dr. Vijay Jagtap Sir, Principal Yashwantrao Bhosle College of Pharmacy for the today's session. With this, now I would like to take the opportunity to welcome our today's guest of honor, the resource person for today, Honorable Dr. Shankar Alikav Sir, in brief. Sir, may I share the PPT? Tushar sir.
No, ma'am, just read it out because might be sir's PPT will be vanished.
Okay, sir. Yes. So today we have with us Honorable Dr. Shankar Alikav Sir. He is M.Pharm and PhD, and currently he is serving at the position of Professor at the Department of Pharmaceutical Chemistry, KLE College of Pharmacy. Sir has been currently working as a professor at the Department of Pharmaceutical Chemistry, KLE College of Pharmacy, Belgavi. Sir has received the Young Scientist Award, that is the Best Young Scientist Award on the KLE University for the year 2014 and 15. Sir has also received the Best Teacher Award from KLE University for the year 2016-17. He has also received the Best Scientist Award, that is the Best Young Scientist Award for the KLE University for the year 2022. Sir has also received the Best PhD Thesis Award by CAHIR for the year 2022. He has also received PhD Best Thesis in Pharmaceutical Science 2022. That is the second prize that Sir has received by D.Y. Patil Institute of Pharmaceutical Science and Research, Pimpri, Pune, Maharashtra, India. Sir has also received a research grant as a principal investigator in the OSTD Chem project entitled Design and Synthesis of Novel Chalcones as Antimalarial Agents, funded by CSIR, CDRI, Government of India. He has also received the principal investigator in the CAHIR project entitled Design and Synthesis of Novel Chlorquinoline Hydrate. Sir has also received the principal investigator in the CAHIR project entitled Antimitotic Based Kinesin Spindle Project Targeting Agents, Design Synthesis and Molecular Modeling Studies in the year 2023. Sir has also received a research grant as a principal investigator in the CAHIR project entitled Multi-Target Based Small Molecule Drug Discovery for Breast Cancer in the year 2024. Sir has publications, that is more than 30 indexed international peer-reviewed publications with 25% Q1 journals. Sir has received 2 Indian patents. He has been invited at more than 30 institutions. He is also a board of study member at 2 different universities. He has been also invited as a peer reviewer for international journals, and he is consisting of professional membership at Indian Pharmaceutical Association, Association of Pharmaceutical Teachers of India, and Karnataka State Pharmacy Council. So with this, once again, I would like to welcome you, sir, and I would like to request you to please take over the today's session.
Thank you, Gauri, for your nice introduction. I don't know how much we can justice whatever you have told nice words. Okay, fine. Can I? Can you see the slides?
Yes, sir, it is visible.
Okay. It is once again, good morning, everyone. Okay, anyone, Gauri or Tushar, you be in line. Whenever I say something, you have to respond. Is it clear?
Yes, sir.
Yes, sir. Yes, sir.
Okay, okay. It is easy because whether we are going right direction or not. Okay, fine. Can we start?
Yes, sir. So,
Yes, sir.
can we just have the slide share mode to the PPT?
PPT is not in a slide share mode?
No, no, sir.
Okay, okay. 1 second. just calm a little. Can you see now?
No, sir, it's not visible.
Visible.
Yes, sir, now it is visible.
It is visible.
Yes, sir.
Okay. Can you see now?
Yes, sir.
Okay. Okay. So if I can you see now? The slides are moving.
Yes, sir. Yes, sir.
Okay. Okay. Fine. Good morning. Okay. So we can start today. There is a one the small topic that is a molecular docking. This is shifting the paradigm in a drug discovery process. Okay. What we could able to achieve today, little things. What is drug design? That is a usually we can call computer aided drug design. You can call computer aided drug design, and some people in a part we are discussing about molecular docking, and in that applications and the case study we are going to discuss today. This is the objective what we are going to we are able to achieve the today's lecture. Okay, can you see this slide, everybody?
Yes.
Yes, sir.
Okay, so I think docking is not only in medicinal chemistry. Docking is in everywhere. Okay, so we can thanks to and congratulations to ISRO team. They have been successfully carried out space docking experiment. Okay, are you agree with everyone? We are last 16th January. Okay, the 16th January, the ISRO has declared we have successfully completed in a docking process. What it means, docking, it is not only to understand the medicinal chemistry or everywhere in other drug discovery. It is in a every discovery part. Is it clear? Okay, so the starting of this note, we can once again congratulations to the team ISRO. They have successfully completed in a space docking. Okay, let us start from the in this good note. Can you tell me who is this gentleman and what you are going to see? It is a, I can say it is an interactive session. Can
Yes, sir.
anyone? Yeah, yeah. Can you see who is this gentleman? Anyone? I think Tushar, you have made a mute to everybody, or how it is? I don't know.
Yes, sir. They have muted one because there might be issue for the
Okay, okay,
mic, so they can chat brother.
okay, okay. No problem. See, if you see this, the great gentleman, he started the research in a different mode. I think he is none other than I think everybody knows green mold. Who is this? This is started is a very important step on the Fleming, that is a penicillin, how he started in a random screening trial and error and method. If you know the story, he just come one morning and he saw there was a some inhibition is takes place in a green mold. Then there is a new era started the synthesis of penicillin and other semi-synthetic penicillin. Okay, how the research as traditional in a drug design, it is a trial and error base. It is a trial and error base. Then if you see in 1940 to 1960, or sometime I can say 1970, the end of the First World War and beginning of the new era of the in a drug discovery, most of the people they are doing like this. Okay, Dr. Arnold Morey, Dr. Arnold Morey, he is conducting some experiment. Okay, If you see early 20th century and late 19th century, most of the research is serendipity or accidental. Most of the research is the serendipity and accidental. That's what 90% researcher they start uh, doing research. What exactly they want to do, they don't know. Okay, but finally they will get some new things that they are telling, yes, we have discovered. And this is 1957, one of the best cartoon. Okay, the how it is in most of the Europe and west part, the people they are giving the lecture over the Dr. Arnold Murray. Okay, fine. Now you may understand research is like this or not. Okay, but research or drug design is not like that. The drug design is a lot of idea or initial leads. Okay, and it is a cycle actually. It is not like this one. It has I have given a 4 people here. Here like a chemist, biologist, and in a spectroscopy person or in a biochemistry person. Then it comes to the drug candidate. If you see how these 4 people can help in a drug research, is the actually it is in a design in a cycle way. It is not in a like a serendipity or you can say any in a accidental. The very important one you can say in a SAR assay, then come to in a different model. All these people they work hard to get initial leads to drug candidate. Initial leads to drug candidate. So whatever we today discussing about in a first part, that is a computational chemist model to prepare the model, and next one is to understand synthesis, to understand the assay, to understand their the structural activity relationship. All these it's actually in a cycle. Each cycle require extensive analytical data sharing, hypothesis capture. Okay, analyzing the data, and analyzing the data. Why I've written it is analyzing the data. Uh, Data analysis is a very important part in a drug discovery. The whatever the data what I'm going to interpret is different, and in front of person is going to interpret is different. That's what the 2 people interpretation you have to take and you have to club. Then you can say there is a new data. Okay, good design needs to access different kind of the data. You can't say is the only one type of the data. We require the experimental data, computational, the modeling data, and chemoinformatics data. So these data we can interconvert one to other. We can interconvert to one other. Then the ideas is going to generate. Then idea is going to generate. That's what most of the time I will ask my student, what is in your interest? Okay, I will tell my interest. There are the 3 to 4 interests, and then they will come one idea. Okay, it is always you have to work with different people idea, and these ideas are none other than they are the same one, but in a different path to reach the some research finding. Okay, uh, now let's start to understand something about what do you mean by computer aided drug design. Usually, most fascinated word is CAD. Most fascinated word is a CAD. Is something to about know and unknown. When I know something, when I don't know something, it lies in between. Okay, what I know and what I don't know. Okay, if I know the protein structure, if I know the protein structure, what I'm going to do. Okay, if I know the protein structure, if I know the ligands, then I can start with a structure-based drug design. It is the most important in a drug discovery. That is usually we can call SBDD. That is a structure-based drug design. Is simple understanding is a protein ligand docking. What you just in a first slide you have seen space docking. So here also in a protein ligand docking. Next come to if uh, I know the structure, but I don't know the ligand, then we can call de novo design. I think Higuchi and Kubani, I think you know the people. These two people, they have worked in a dam to understand what do you mean by de novo design and other things. When I know the structure, but I don't know the compound, how to design. That is a usually we can call the de novo design. Then come to I don't know the protein structure. I'll give the one example. The simple example is nowadays in a Ebola. Okay, can you see what is the structure of Ebola? It is still question mark. Okay, it is very difficult to understand in a first phase of corona. Okay, the people they don't know. Still, we can call NS. What do you mean by NS? Non-structural proteins. The very important to understand when we don't know the protein. When we don't know the protein, then the research starts. It is like in a ligand-based drug design. It is called ligand-based drug design, usually LBDD. I think QACR you might have heard, everybody. The QACR is a part of LBDD. That's what, if you see the corona, most of the people, they used anti-viral agent rather than anti-corona agent. They used antiviral agent. Most of the time, these antiviral agent and other agents, especially aminoglycosides, they have used a lot of these compounds. They come to know whether these compounds can going to inhibit the in a corona protein. But fortunately or unfortunately, we have succeeded with the vaccine. And once again, we have to congratulate the vaccine provider. And I can say why I use the word congratulation. Usually the research starts, it's more than 10 years and almost 1.5 to 5 billion US dollar. And I can thanks to Indian authority and ICMR collaboration. People, they have come with a vaccine within 54 weeks. Within 54 weeks, it is, I think, less than or equal to 1 year. They can reduce the 10-year pain and the cost and other things. That is because of so many people directly or indirect work. Then that's why it is comes to difficulty to structure-based and ligand-based drug design is if I know the ligand one or two, then I can go to the similarity search. Okay. If I know several ligand, then I can go pharmacophore search. Okay. Then if I know more than 20 drug, then I can go with QSR. If I know more than 100, then deep learning, machine learning. I think everybody knows. In other time, whenever the time comes, I can explain you what you mean by deep learning and machine learning. Deep learning and machine learning is nothing but an extension of, or it is a next part of the QACR. That is a quantitative structure activity relationship. Then come to is the very important things. If I don't know the structure, if I don't know the ligand, then I have to visit either biochemist or pharmacologist or biotechnologist. Need experimental data. Need experimental data. I think most of the people, whenever the computer-aided drug design comes, this slide is the most common in a different format. I think I took little time to understand here. Now let us start. One is the SBDD. Okay, that is a structure-based drug design. Can you see this beautiful girl? Can you know the anybody? What is the story behind this beautiful girl? Yes, Tushar or Gauri? No one?
So Cinderella.
Very good, Cinderella. Okay, so it is just Cinderella is the one of the best example for the drug research. Okay, what happened after the in a night in a morning bell? Okay, she left the party and the king is searching her. Okay, but she has kept something tail behind. That is her in a sandal or shoe. Next day, the king has ordered you check all the beautiful girls they are in a present in my city and where this sandal is going to fit for her. Okay, so now so this is the way of drug design. I know the compound, but I don't know the where is going to target. It is once again one of the best story. It has been written in chemistry and biology in 2004. From this research, Cinderella research, or Cinderella, to understand the Cinderella, is a very important in a drug research. That is, they are going to check each girl whether you are the right in a girl which our king has saw in a night. Okay, same way, target is identified, where is a ligand? Target is identified, where is a ligand? So I know the ligand, but where is the target is the vice versa. But to understand this, the modern drug research is nothing but a fishing. Is modern drug research is a fishing. More or less people they are understanding target identification in drug discovery. Can you see this person? He is just like a fishing. He want to understand good what I can say in a fish, but he has got the some other. Okay, that's usually we can call fishing is nothing but a drug discovery. Fishing is nothing but to identify the good research, to identify the good target, to identify the better compound. Then for this only, I have applied in your docking. What I done first with my knowledge, I have sketched some structure. I have sketched some structure. Then afterwards, I have done the modification. I have done the modification. So once these two things, most of the people they understand if I know the software, so I can do the research. No, software only it helps where you are going wrong. Software it helps where you are going wrong. Okay, the design is in your intelligence and your experience. Okay, so that's what I have given simple one in a fused ring. That fused ring I have applied in a two functional group. Okay, one I made a star, other one I have done this one. Uh, if you see this, then I have taken in a target identification. Then I have done in a drug, vehicle, drug candidates, so many things. Afterwards, when anyone this my modulated drug it has best fit, then I immediately I pick it up. Can you see the hand? I have picked it up immediately. Is it clear? Okay, are you understand this? Can I? Is it the right way, or I'm going fast? Tushar, Gauri, is it okay?
Yes, sir. Yes, sir. Okay, no issue.
Okay, everyone, fine. It's okay. Now this is the first part of my research that people they will understand only if I am having software I can do the research. No, without software also we can do the research. The people has to ask for their mentor. People ask to their yes. Any question?
No, sir. For question answer, I have just switch on the mic, but I think I need to mute it.
Okay, fine. No problem. No problem. Okay, then to understand the structure-based drug design, we require two things. Yeah, please unmute yourself. Anybody, if you're okay.
Yes, sir. Done, sir.
Okay. So when I want to know the structure-based drug design, let us Tushar, we are concentrate only one today. That is a structure-based drug design that is called SBDD. Okay, that is usually we can call in a molecular docking, or the best way is to understand the interaction between the two molecules. That is a big molecule and small molecule. To understand the structure-based drug design, we require the two things. One is molecule, another one is the also molecule. Then you may ask, sir, why you are telling the two molecule? One is the big molecule, another one is the small molecule. In a big molecule, when I know how to start the research, always the people we have to start with research with a co-crystal. With a co-crystal. What do you mean by co-crystal? It is just like a inhibitor, and I know the binding site, I know the active compound. Then second one is I don't know the in a binding site. I don't know the binding site. So these are the two things what are where we are going to understand the in a structure-based the drug design. Okay, if I want to start the structure-based drug design, okay, then usually the one important things in that molecular docking. One important things in a molecular docking. In a molecular docking, we can understand, we supposed to understand what is the search algorithm, what you mean by score function, what do you mean by reverse docking, what do you mean by forward docking, what is mean by in a blind docking, what do you mean by in a virtual screening. To do all this, we have got lot of multiple in a free software to pay software. Okay, there are the very important softwares are available in a Autodoc is there, Autodoc Vina is there, Gold is there, Doc, FlexX, Autodoc Vina, Rock, ZX, Glide. There are so many, PyMore, RC Docker. There are n number of docking softwares are there. The one should understand which in a docking software I'm going to use. Okay, before I'm using the docking software, whether I know the receptor, whether I know the ligand. That is very, very important. If I know the ligand, so then I have to find out which receptor is the very important one. If I know the ligand, then it's a only to understand their pharmacophores. Then if I know the pharmacophore better, then I can make an alteration of the pharmacophore to understand all these things for the help of the to understand compounds using the molecular docking. Okay, next come to understand this molecular docking, just go a little about history regarding the molecular docking, where we are there. Actually, it is the way back in the 19th century only. Okay, there usually we can call medicinal chemistry starts, and each key is going to open its own lock. There is a key and lock system. It has started, but it has taken good importance over the late 20th century. That is in a 1980s, we can start in a match algorithm. Usually can start in a rigid docking. Then come to 1986 to 1990, it is a semi-flexible docking. And after 1994 is a more or less flexible to rigid to flexible docking. Okay, it is 2010 onwards, you understand machine learning, deep learning. Okay, it is a very, very important to understand. In a 2020, after the people, they are understanding virtual screening, okay, and cross docking. Virtual screening also was earlier, but from 2020 onwards, because of the COVID, people, they are going to a lot of these the virtual screening ideas. Okay, then come to the typical. If you want to anybody, it is a very important note down. If anybody want to docking, okay, it is first one, the person who want to do the docking, he should know the target. Second, he should know the ligand. Okay, the first one is target selection. Second one is ligand selection. If I know the target, then you have to prepare the target. If I know the ligand, I should provide the proper ligand. Okay, then we can do the docking. So once again, after the evaluation docking results, you have to go back. This is like a cycle. This is like a cycle. Fine. But what are the different types of the docking? I won't take the too much of time. Reverse docking and normal docking. These are the two important things. This one, reverse docking, is much more present in the last decade, but the people, it has been very well established after COVID. That is the new word, drug repurposing. Okay, the drug repurposing, the new word, it has started after the COVID. If you see 2020 and 21, billions, I'm using the word billion, not million, billions of article they have published in a scientific, but I don't know how much article they have given in a data in a scientific world. But docking is the first we can understand the protein of your interest. If I want to do the simple in a anti-tubercular, then I have to see Mycobacterium smashmatis or Mycobacterium tuberculosis. If I want to work in a gyrase, the one should know the topoisomerase 1, topoisomerase 2, or 3 or 4. Okay, if I want to work under the anti-cancer, then if I know the kinase, if you know the kinase, there are the plenty. If I know the VGFR, EGFR, ABL kinase, CDK kinase, the PARP kinase, JNK kinase. There are so many. This one, that is nothing but a protein of interest. If I know the protein of interest, then one should know the molecule. One should know the molecule. That is nothing but his protein of interest. You can put into the most of the in a PubMed is there, Zinc database is there. Okay, PubMed Central. There are so many the free in a what I can say software or in a websites are there. You can find out how to choose the molecule and other things. When you choose the molecule, you can download. You can download either from the drug bank, either from the Zinc database, either from the PubChem, or in any one of these what above I mentioned. So once you complete it, then you can see the binding of the ligand. Okay, this is the way. So reverse docking, it was there in the last decade much. If I'm having the one compound, okay, like everybody knows one drug, it can treat multiple in a disease. Then that drug is the called we can universal drug. Or most of the time after COVID, the people they are finding most of the antiviral drug they can use in a specific coronavirus. They can use the specific the coronavirus. If you see this way, one drug and you can use the multiple target, then you can get the possible in a binding protein. So this is not in way it is very well established in a future medicinal chemistry in a 2014 only, rather than in a if you see drug repurposing and other things is the most of the things in a 2020, but it is a one of the very nicely explained in a last decade. That is a if anybody interested, you can go the future medicinal chemistry in a 2014. He understand how the docking system and how the reverse docking system it works and what are the things they are present and what we required to understand. So once this know in a reverse docking, or you can call it virtual screening, or you can call in a different word, then in a artificial intelligence or nothing but AI assisted reverse docking, or now we can call AI assisted machine learning, deep learning, and the similarity search. Okay, so if you see this, it has been well established in a last decade. It has been well established in the last decade. Why we want docking? My most of the time, my this question to my student and other things. If I want to start any one chapter, it is my first question. Why should I study this chapter? What is the outcome? Okay, that is a very, very important. Why should we go for the molecular docking or whatever the structure-based drug design? Answer is here, either reverse docking or in a molecular docking to understand target identification is very, very important. To understand the target identification, I want to do molecular docking. Okay, to understand the side effect and toxicity, to understand the side effect and toxicity, so I can do the structure-based drug design, or in a if I want to do in a ADMK or other toxicity studies, if I want to perform, then the structure-based drug design or molecular modeling, or when I say in a computer-aided drug design is important. Drug repurposing, already I told you. Okay, then drug target network development and the receptor design. It is a very, very important. Everybody knows like Uniport. Uniport is one of the highest whatever the in a sequence, protein sequence, it has been available in a Uniport. The students are most of the researcher, those who don't have any the specific target, they can start with a like a Uniport, then they will go with a different homology modeling, then they will come to whether is there any nearer to in a protein data bank, then they will start with their research. So why I'm going to do docking and other things, it is because of to target identification, to reduce the side effect, to understand the exactly mechanism of action, drug target network development, and receptor design. Okay, to understand this, we are having there are n number of docking software, but whatever I mentioned here, these are the some of the docking softwares you can believe, because Dock, AutoDock, FlexX, Gold, and Glide. Here, Glide, Gold, FlexX, Dock, These are the commercially available, and Autodoc and Autodoc Vienna is the freely available. Okay, when I know the docking is important, how to perform the docking? There are 3 to 4 important steps in a docking, or anyone, if anyone you are using the in a Autodoc also, that is a ligand preparation. Ligand preparation is very important. What do you mean by ligand preparation? In a ligand preparation, you can see I will tell you small story. I think most of the time, whenever I'll start my research in a computational study, so I can tell this around 30-second story. You know, ligand preparation or protein preparation, you have to make the compound should be minimized. The compound should be minimized. Okay, Tushar, Gauri, you are here. Hello.
Yes, sir, you are audible.
Yes.
Okay, fine. I'll tell you, Tushar, you just coordinate. What do you mean by energy? Okay, can you see the in front of your father, what is your energy? And in front of your mother, what is your energy? Can you tell me? In front of your father, your energy is less. Yes or no?
Yes, sir.
Okay. In front of your mother, your energy is very high.
100%.
Okay. So, but in front of your best friend, okay, your energy is minimized. Your energy
Yes.
is minimized. So that's what energy minimization or protein preparation is a very important in a molecular docking. The first step, everybody, you should know you have to do energy minimization, and there are so many free softwares are available. Even if you are having in a Chemdra is one of the best software to perform the energy minimization using the Monte Carlo. There is a calculation, that is a molecular mechanics they are using. Okay, if you are using, if you want to go with online, there is a ChemExon, one software is there. If you ask for the student evaluation, they can provide you. Okay, Marvin Sketch and Marvin Draw. These are also another. There are other free softwares are available, or you can upload your compound and it can minimize. The first one is the ligand preparation. The second one, the same way in a protein preparation. Okay, second one also is the same in a protein preparation. Okay, same in a most of the time protein. How to select the protein? You have to select the protein in a such a way it should be in a X-ray crystallography or X-ray diffracted in a structure rather than in a NMR or cryo. If you won't get any X-ray diffracted or analyzed or identified, then you have to go NMR and other this one. Then third one is the very important one is predict the best ligand binding site or your compound binding site. There are so many different software they are having the different, but Autodoc you require the whole doc first. If you know the ligand, then you can use the that where the ligand has bind have a good bind. So then best sitemap is one of the best in a software in Schrodinger. Okay, you can use the sitemap and immediately it can tell about where exactly in a binding pocket is there. Why I'm telling this? Because most of the protein it containing the shallow binding pockets or hollow binding pockets in the upper where. Okay, these shallow binding pockets, it looks like a in a binding site, but not exactly in a binding site. Then come to the docking parameters. So in a docking parameters, one should know what search engine, what search algorithm you are going to, whether you are going to in a genetic algorithm or what, how it is you are going to understand. OPLS is one of the best in a force field what we are going to use in a in a lick prep, sorry, in a glide, and there are so many other field forces are available to understand the in a docking and docking process. The people they will understand the only docking this much, but actually the work starts after the docking. That is called the analyzing the docking the result. the analyze docking, the best analysis or best analyzer who can make a best in a lead compound or docked compound. Okay, so this is the best way, and even in a protein preparation and other things, these are the best way to understand even in a reverse docking, or it is little bit different in a reverse docking or drug repurposing, or I can say cross docking. Here only the two steps are important, or they are same, ligand preparation and protein preparation. But the earlier what we have done, the multiple compound one target. Here in a different multiple target in a one compound, multiple target in a one compound. Then other part will remaining saying, okay, preparation of docking parameter, docking studies, and then come to analyze the docking result and lead or possible the protein, what we can say. So now for this in a molecular docking, or when I say something, it can very easily to understand in a small molecule. How this small molecule and how this big molecule can you take? Okay.
Sir, your slides.
One second. I will open how to fetch this. Okay.
Okay, sir.
Whether my window is visible or not?
Not yet, sir. Just
Okay.
share full screen, sir.
1 second. How to do that?
Sir, after clicking on share, there is a left hand side. There is an option of screen.
Okay. So
yes.
Can you see now? Can you see now?
Yes, sir. Yes, sir.
Okay. Can you see protein data bank PDB? Can you see the slide?
No, slide is yes,
No.
yes, sir. Now, yes, now it's switched.
It's a RCSB. Just note it down if anybody, if you're www.rcsb.org. Okay. So this is the one site where you can get a lot of information you are going to understand. Okay. The person, those who are working in a different compounds, just I can say anti-tubercular, most of the people anti-tubercular and anti-cancer and anti-diabetic, the work they are doing. So I can just put like a 4B 6C. There is a one simple what I put in a PDB ID. Okay. It is like a protein data bank or 4B, how to collect or how to download or how to use the big molecule. These are part of the big molecule structure of Mycobacterium smegmatis, gyrase B ATP domain complex with amino pyrazinamide. Okay, this is the how we can understand one this. Then come to, if I know this protein data bank ID or PDB ID, it is usually the unique 4 digit. It include the numeric number and in alphabets, both it can include. And then if you go to the experimental data, so you can get like a X-ray diffraction and resolution, then come to R value, then you can see. So next one you can see, you can choose only in a X-ray diffraction. There is no any, if you take other one, you may have some, you can get that result, but that results, it is a definitely in a bias or sometime questionable. Okay, if you choose X-ray diffracted one, if there is no X-ray diffracted proteins are there, there is no any other way, but you have to go with the available data. So once this you can complete, then you can understand what is the structure or ligand interaction. You have to go the left click. This is the one you are having in a protein. In that you have to click in a ligand interaction. In a ligand interaction, you can understand whether what is the type of the ligand is there, how the ligand interaction will takes place. All these things we can understand in a. Okay, this is the best way to understand in a ligand, this one. And here, this just you can uh, rotate and come exactly where you can understand the something in a ligand and how it is uh, uh, fixed in your the pocket. Okay, so once this complete, you can just move around and where exactly it is the bonding is takes place and other things. Just very important, if wherever I made, there is a in a down right hand side, there is a formation of hydrogen bond. Okay, the glutamine 41. Okay, just move like this wherever the it is possible. Okay, then you can see the interaction and how the interaction is available. Okay, what type of the interaction is available. All these things will come to know. In amino the pyrazine or which compound where it is the targeted is there, which water molecule. Can you see why I'm telling you? Most of the autodoc people, they will remove the water. Okay, but I can say water mapping and water is a very, very important in a nowadays because it makes the bridge between your compound and in a molecule of the big molecule. Okay, if you want to know only the ligand, then you can see how the this ligand representation, the ball and stick. If you want to put, you can change this one. But if you see these structures, how exactly this structure looks like, how exactly structure it looks like. Can you see it is just like a half U or L type. Okay, half U or in a L type. So one understand here, my compound should be in a such a way, it should be flexible or it should be have a nitrogen or it should be a 5-membered or 6-membered compound. And these minimum things required to if I want to do mycobacterium smashmatis as a my target. Okay, so once this target, if I know, okay, how to understand, come back in the original this one. Okay, then we have to see is there any structure in a missing and other things. That information you are going to get. So here you can understand which molecule is there, how this molecule interaction will be there. Okay, what type of the molecule you can understand in a 2D structure. This 2D structure, why you are supposed to you can, then you have to understand whether my molecule is going to have a, why I choose this only. Then you should choose the protein someone has worked already. Already this molecule, this protein, and some people they used, and IC50 value also is there. It is a quite good, that is a nanomolar range. IC50 value is there. Then you have to choose such in a target. Okay, so now it is what I told in a, to understand anyone in a, to understand which target I'm going to choose for the antitubercular activity. If now then come to where the molecule I'm going to select. Okay, it is a best one is drug bank. The drug bank is there is one of the best. If you just put in a ciprofloxacin. Okay. So next, the ciprofloxacin come and it can tell a different way how this, whether this antibiotic treat wide variety of the infections in the body. And even you can download. You can download in a different like a mol file, SDF file, PDB file. The most of the time you can go for the smiles. Okay, what do you mean by smile? If any other time, if I can explain you, it is a smiles are very, very important in drug research. That is simplified molecular input line entry system. I repeat, okay, the simplified molecular input line entry, the system. So these are the system. It is a very important for the in a drug research, or especially those who are doing the computational study or the chemoinformatics. So now can you understand where we can get the small molecule and where you can get the big molecule? Okay, once you know these things and how to download these, there is options are there in a download options. Always you can download in a PDB format. Okay, you just click in this one. Can you start? You can see the PDB, that 4B 6C. You can download it and you can save in your wherever the work you are doing. And this way you can use this protein data bank file and in a protein that is a drug bank, and even you can use PubChem is there. There are so many Zinc databases there. There are so many other databases are there. You can very easily download and you can do this one. Okay, can you come once again? Come back. Can you see the slides now?
Yes, sir. Yes, sir. Just again put it in presentation mode, sir.
Okay, now I have put the presentation. Can you see now the small molecule like this?
Sir, there is one issue. Okay, click on display setting.
Okay. Fine.
No, or on the right, left corner.
Left corner. Okay. Now, whether you are can you visible the slides?
Yes, sir. Slides are visible now.
Okay. But not in a presentation mode?
Yes, I'm not in presentation.
Can I do the presentation? Okay, 1 second. Can you see now?
Yes, sir. But again, there are two options we are observing over here. Sir, just above your slide, na, there is one option, display setting.
Display setting. This?
Yes.
Okay, I don't know.
Okay, sir. That nearby that three dot, na? Nearby that three dot. Just click on that monitor window.
Monitor window.
Okay, let it be, sir. Sir, it's now again. Yes, we just instead of wasting time, we just continue like
okay.
this.
No, it is going right now. No problem.
Yes, sir.
Okay.
Yes, sir. No issue.
Fine. Display. Okay, can you see now?
Yes, sir. Yes, sir. Now perfect.
No, perfect. Okay, fine. Okay, so if you see this one, okay, then I have to select first if I want to work in a anti-diabetic. Okay, so what is my interest? I have to see the small molecules or which molecule is there in that. Okay, so now if I'm interested, the student or researcher should know the in-depth literature search. Okay, what type of the molecule they are present in the market. Okay, rather than more than 10-year-old, if you see, and what are the presently available in the market, both they can search. So I'm just done this. Can you see in a veldagliptin, pioglitazone, okay, metformin and ercarbos, glimepiride and gliptins, and these are presently available in a market. Okay, but from this, I want to make any one design. Okay, I will go just now in a some where we are docking is how is help and how we have proceeded with in results. Okay, from the help of in a literature search, I have come to understand, yes, I want to do work under the one umbrella, that is thiazolidine dione. Okay, if I prepare thiazolidine dione, it is researcher duty to understand what are the different thiazolidine dione moieties or thiazolidine dione compounds are available. This can help the researcher and student to design the molecule. So once they design the molecule, then if I design in the same molecule, then it is not a good. So I have to take the two part. So one is here I took the in a glyphosate, I took the pioglitazone. So what I done, some part I took from the glyphosate and some part I took the pioglitazone. It is a more or less me too drug, or I can say in a re-research, because we are working in academic section, academic setup. We don't have in a high-end research in a what I can say facilities. Okay, only whatever the available things, each person you can start with. So we have designed the molecule with the help of available research literature data. Once if I design this molecule, my dear friends and my dear student and my dear the fellow researcher, first you sketch the final compound. First you understand what is the final in a active ingredient you are isolating from the extraction. If you are in a pharmacology person, if you are in a pharmacognosy person, or if you are natural chemistry person, people without knowing they will start extraction. First you should know whether glycoside, whether in a protein, in a terpenoids, triterpenoids, whether they want to extract is a very simple flavonoids. Okay, but the person should know what is the final structure and what could be the nearer to final structure. Then only the molecular docking or this computational chemistry helps you better way. Okay, from this way we have searched some more in a literature search. What could be the common we have find in our designed compound, already published compound? So then we have gone with the most of the national and international journals, and we find out there are so many similar compounds. They are already published and they have shown they have elucidated the structure of the anti-diabetic activity. Then we have started making the like a retrosynthesis. Retrosynthesis is nothing but how to synthesize the final compound. Then we have done in a different formats. Okay, then once the molecules ready, then you have to choose the your target. So here target is here pioglitazone. What I told, listen everybody is carefully. Once I targeted my compounds looks like in a pioglitazone or my compounds, I'm going to target in a PPR gamma. My compounds are anti-diabetic which inhibit the PPR gamma expression. Okay, so then you should know my compounds either in a nearer to pioglitazone or oxyglutazone, balaglitazone, or they are better than the existing drug. When you know the compound, then how the compound is existing in a space actually in a drug target. So then you can understand this is the way what are the amino acids are responsible. The tyrosine, histidine, and serine. These three are important for the structure of the compound. That is a tyrosine 473, histidine 323, and serine 289. These are the compounds or these are the amino acids is responsible for the activity. Then I have to dock my compounds. So compound E and compound F, somewhere I have given the name, and these compounds, the 1 second. Sir, I am on the guest lecture online. Okay, so there is a my compounds I have in a dock, and the most of the compound they shows better in a binding activity, but some compound they shows similar like a tyrosine 473, histidine 323, and serine 289. So these these amino acids are responsible. What I told, what I've seen in a target compound or in a main compound, whether the same interaction or same binding site my compounds are fixing. If they are fixing, then I have made a finalize to synthesize these compounds. From this, we have outcome is we are able to publish in most of the given journals. Okay, then come to the when these results can make us to a better way to understand kinase, to understand. I'm doing this study from last couple of years to understand what exactly in a kinase domain. To understand the kinase domain, there is a N lobe and there is a C lobe and hinge region. Methionine 790. The people, those who are working, I'm requesting to go through in this gatekeeper. Okay, this gatekeeper is a very important in a methionine 790, which can make your compound is the very active and other things which can you can prepare or you can design better in a kinase or in a tyrosine kinase inhibitors. Okay, apart from this, when I want to do the tyrosine kinase, the one should know once again literature search. How these literatures, it's most of the anti-cancer drugs they have started in a 2000 and it almost very important in up to 2020. If you check the first generation, second generation, and third generation, more or less nitrogen heterocyclic is the most common in there. But if you check in a first generation, there is a fused compound in a quinoline or quinozoline. Okay, but if you come to in a third generation, they have shifted to in a different in a fused compound along with nitrogen, either the pyridine, pyrimidine, or some other in a hanging compound. Okay, these are important to understand. And afterwards, we once again gone through in a molecular docking. We understand leucine, valine, alanine, and methionine. Leucine, these are the most important amino acids. If my compounds are bind to in this section, so I can say my compounds are good. Apart from this, we finalize the even serine 797 amino acids binding and methionine 790 binding. These are the very, very important. Then when we go the cross-checking, gefitinib, afatinib, and the other tinibs, these most of the kinase, they are having the same in a binding site or binding domain. Then we started analyzing the small molecules, how the small molecules they are generated along with these design. Then we started the different compounds, and we have synthesized more than 50 compounds. Along with that, we understand the different pockets, and then come to we are synthesized some drugs, and we could able to publish in a Q1 journals. Even we are little bit changed the here. If you see, we have done in the ortho position of the hydroxides. We have just shifted to in a para position, and we could able to better the result. It can have IC50 value just in a 1.9 nanomolar to 2.3 nanomolar. We could able to the publish this paper in last year. Okay. Apart from this, now next our research we are going to the small molecules to new molecule. The mutant, I can see methionine 790 now. Methionine 793 is a very, very important amino acid that is called mutation or other things. Lead optimization and other EG5 or in a EGFR, we are going to understand some of the compound we able to synthesize. They can show us better result in IC50 value. They are much below the 10 or this one. Okay, I can tell you other story. This is the Mycobacterium smegmatis. Okay, in a reverse docking or in a virtual this one. Same what I shown you, just 4B6C. In most of the people, anti-tubercular activity or anti-tubercular, antimicrobial people they are performing, but they have to go through these things. But first, they have to analyze their compound and how it is matches. Okay, if they want to do in a Mycobacterium smegmatis or Mycobacterium tuberculosis, the compound should be in a aspartic acid 79. Okay, it should interact with in a aspartic acid 79. Then only they can go for the synthesis or other one. Arginine also, there is a pi to pi-cation interactions. And to understand this, one should know amino acid sequence and what are the amino acids. There is a single line quotation or 3-line quotation or this one. Then now the people, just I'll come back this slide. The people they want when they sketched or when they have multiple compound, even they can go with the machine learning and the deep learning. Okay, for that machine learning and deep learning, the need for study the any student is very, very important. The need for study is these are the different type of the cancerous. Which cancer I supposed to target? Which cancer I supposed to target? That is a very, very important to understand in a present study. And then you can have a different compound case study, and we are able to publish in a different platform. This one case study is the very, very important. Okay, we don't have the much about in a biological data, but we able to prepare these compounds according to the literature search. Afterwards, we started fishing target. Then these are the so many targets are available according to these compounds. So finally, we'll come to know with reverse docking. These reverse docking, we can come to know acyl enoyl acyl reductase is one of the best compound. Then after this, we have gone through in a enoyl acyl reductase in depth understanding. We have performed the antitubercular activity. Fortunately, there is a same compound. We can see in a 5B and 5E. They are the compounds, and even 5P. Most of the compound, those who shows better results, they have also provided the better result in a biological data. So afterwards, we can understand the correlated with these study with in a computational. Once again, it has proved we could able to publish in this one Chemistry Africa journal. And another case study we could able to make in a better the small molecule inhibitor, that is a DNA gyrase inhibitor. We could able to publish it is a journal of biomolecular structure and the dynamic. Okay, most of the people, this is the one study I can tell you, people, they will understand this is my chalcone, but I can say instead of chalcones, you are preparing the hydroxy compounds. Most of the people, they will prepare the chalcones in a chemistry people. They try to understand whether your prepared compound is right or wrong. Why I'm telling, okay, the actually you have done the docking of this structure, but you have sent the activity of this compound. I'll repeat once again. You have docked the compound like this, but end up with this compound in a synthesis. And this by this time, your docking result and the experimental results are not going to match. This is the once again I can say limitations. I can conclude this overall what we have done in the last 2 decades. We have synthesized more than 300 small molecules. Okay, we have targeted in anti-diabetic, anti-tubercular, and anti-cancer activities, and few compounds shows they are in a better activity, but only in a handpick, only handpick compounds they can become a lead molecule, and for this we could able to make better understanding the compounds and better understanding the targets. Okay. More than 20 international indexed paper with Scopus and Web of Science. And these are the some molecule we could able to achieve with our student. They can less than in a standard drug. If you see, these are the just nanomolar range, 3.9 and around 6.7 nanomolar range. These compounds, they are better than gefitinib. We are planning for the future for these drugs. Take-home message is there to understand the research. One should know in vivo results, in vitro results, and these in vivo and in vitro results he has to compare with the in silico. Then he can get the better. Okay, I'll tell you another this one important message for the research. It's required the teamwork. It is not a one person. You have to believe the others and others data, but not like a teamwork. It is also teamwork without dialogue or without this one. They are in a different track. Okay, so teamwork it should be in a with the dialogue, with taking the consideration of everybody in your group, and then you can achieve the betterment. This is the just my one of the existing my people like Madam Dr. Preeti. She is completed and now she is having its own group. He's Mr. Shankar, he's Rohini and Vaishnavi and Ram. These are the some the PhD students, they are working under me. Okay, thank you. If any thing is there. Yes, Tushar, way to you.
Yes, that is. Dushak, ma'am.
So,
sorry, I'm not hearing.
Ma'am, your voice is breaking.
Yeah, Tushar.
By giving an example of yes, sir. So I would quickly like to summarize the today's session.
Okay.
So in this today's session, sir, we have been given a detailed information regarding molecular docking. So firstly, he spoke about space docking experiment by giving us an example of ISRO. Sir also spoke about traditional drug design, which has been considered trial and error based. Sir also spoke about the details of computational chemistry and CAD. In CAD, sir spoke regarding known protein structure and unknown protein structure, that is In known protein structure, he also told us regarding structure-based drug design and protein-based docking. In unknown protein structure, Sir spoke about ligand-based drug design and QSAR. Sir also told us that more than software design plays a only gives us the information whether the process is right or wrong. Sir also spoke about details of drug design and molecular docking. Molecular docking, it is a technique which is used to identify the potential drug candidates. Sir also explained us a typical docking workflow and docking programs like dock, autodock, Volt, etc. We also saw the use of RCSB protein data bank. Sir also spoke about the importance of targeting the small molecules. And he also told us regarding the case studies that were performed. So thank you, sir, once again. And now we would like to move on towards the question answer session. If any participants are having questions, you can please raise your hands. Or you can also put the questions in the chat box.
Ma'am, there is one question in chat box.
Hello.
Yeah.
Good morning, sir. It's really very
Good
pleasant
morning.
listening you, and very meticulously you have thrown light on so many things. And really, I would love to listen this video from time to time, or many a times, I could say. I have a query, sir.
Okay.
Sir, would you please throw a little bit more light on reverse docking and polypharmacology? Are
Okay.
they same, or is there any, or what is there, like minute things which we
Okay.
might not be knowing?
Okay, madam, this is a very good question. That's what this reverse docking or polypharmacology, it is a very last 2 decades was there, but it has been well documented after the post-COVID, because the people, they are searching one drug in multiple targets. Okay, but it was there in before this one also. Polypharmacology or reverse docking, one or other same. The polypharmacology people, they want to say one extract, it can work in a multiple target. But it is exactly opposite of the people, those who are working in a specific target. They will tell, if I want to do the specific target, my compound is going to, or my extract, or my API is going to target in other enzyme also, it can elicit the side effect. These are the two class of thoughts, or two people having, but I'm going in a both multiple target and as well as in a specific target. In a multiple target, if you go, you can reduce the dose. Okay, the single target means you can increase the dose. Okay, nowadays, you know, the cancer or diabetic or these, you can take the very long period of the medications. Okay, then come to your question, madam. The reverse docking is nothing but in a drug repurposing. Reverse docking is nothing but a drug purposing. Suppose I'll tell you, you are having one drug in a paracetamol. Now I want to take in only to reduce the elevated body temperature. Okay, I'm having all the data. Okay, suppose if I want to use the same paracetamol for to reduce the glucose. Okay, then it is a good for me. I don't want to do any preclinical studies. I don't want to go any approval to this one. Directly, I can launch in a next day. This paracetamol can also reduce the blood glucose. Okay, suppose this is the one way, but whether these compounds exactly matches of the existing pharmacoeconomics. What I mean to the pharmacoeconomics, if any one drug, if it is I can say metformin. So metformin is the basically one of the wonderful drug. How this people they work on, if my compound is better than 15% of existing drug, I will repeat, minimum 15 to 20% better than existing drug, most of the people, they will go for the next level. This is the one. Second one, the 50% less toxicity from the existing drug, then that drug will go for the next way. So all these things, it is going to help in a reverse docking.
Thank you so much.
Okay.
So we have one more question from Rakshanda
Yes.
Korzwekar ma'am. She is asking, can QSAR be used to predict activity of designed molecules with the help of activity of known compound?
Yeah, that's what I told. You can predict, but you should have lot of data. The people I have seen, they have done a QSAR having the 10 compound, 20 compound, okay, with having the only one R group, then you may get false positive results. Okay, you can predict, but your data bank should be very high. Minimum, I can say, if you want to predict unknown compound activity, the known compound activity more than 100, or I can say more than 1,000 if you want to do machine learning. If you want to QSAR, at least 60 to 75 compound data, it should be in a wide variety of or diverse compound data. If you are having 100%, you can predict in an unknown compound.
Yes, sir. Tushar, sir, are there any more questions?
Just give me a couple of minutes, ma'am. Just checking on YouTube also.
Okay, sir. So we also have with us the principal of Yashwantrao Bhosle College of Pharmacy, Dr. Vijay Jagtap, sir, for the today's session.
Okay. I think we have managed the time. No? That's what yesterday I called this one. What type of the audience and other things. I have to reframe my talk and other things.
Yes, so the time is perfectly managed.
Oh, okay.
Yes, sir. There are no question from the YouTube also. Okay.
Okay. So once again, thank you.
The time management.
So I would like
thank you.
to.
Sompla madam.
Yes, sir. So I would like to take this opportunity and thank you once again for having such an amazing session for today. You have been an amazing resource person as well as an inspiring teacher for us always. Also, I would like to thank honorable management members of Yashwantrao Bhosle College of Pharmacy and Bhosle Knowledge City, Principal of Yashwantrao Bhosle College of Pharmacy, Dr. Vijay Jagtap, sir, for allowing us to conduct this session. Also, I would like to thank all our dear delegates for their patience listening for the today's session. Thank you so much.
Thank you.
Thank you, sir. Yes, we will.
Thank you.
Yes.